Hey there. So we're looking at starting chapter 18 today. This is going to follow and go in much a similar path to what we just did in chapter 17. We're going to look here a little bit more specifically at how smaller groups can change and evolve over time. So we're looking specifically here at population. So this is evolution of populations, and we're going to try and focus on some of the more specific details of how these processes happen. All right. So go ahead and get the title for chapter 18. We are going to start in section 18.1, which is about genes and variations. So what we're going to look at here is specifically where does variation come from? What creates the uniqueness that we see among many individuals in a population or species? And then how does that variation change, over time? All right. So as part of this section, we have three major objectives. So we're going to look at evolution in terms of, what do genes have to do with it in terms of, how do we inherit traits? And based on that, how can that inheritance, change what's being selected for in a population over time? Again, we're going to talk about where variation is produced, where it originally comes from. And then we're going to look at some other factors that can determine how phenotypes or physical appearances are changed based on genes and some other factors as well. Okay. So these are terms that we've used in the past or potentially not, right? If we're looking at environmental science, we've not used these terms yet, but I have included definitions for us. All right. So even if you were, in bio and you went through these and you've forgotten what they represent, here they are again for you. So genotype, again, refers to the alleles or the variations of a gene that we carry on a chromosome. When we talk about genotype, we're really representing the symbols or the letters that we use to show which versions of a trait are being passed down. So I've got examples there like big A, big A, little a, and little a, little a. Those are all representative of genotypes, okay? When I say phenotype, if you remember what that's referring to is what you actually see on the surface, right? Like the physical appearance, the expression of a trait. So in other words, genotype is what you actually inherit. It's the molecules you get from your parents. Phenotype is how you take those molecules and show them to the world. Okay, so phenotype, think physical appearance. Genotype, think genes, chromosomes, and molecules. Okay, so when we talk about natural selection and evolution, we're really looking at the impact on a phenotype. So nature doesn't know what copies and what combinations of alleles you inherited from your parents. All that nature knows, and I'm using that term loosely here, but all that nature can select for is whatever it can see, right? So whatever traits are actually being shown is what we would call our phenotype, and that phenotype is what's acted upon by nature over the course of natural selection and evolution. It really, genotype plays a role in that, but in the broad scheme of things, the piece that's going to matter the most is going to be that phenotype component, okay? So when we talk about variation, we're really looking at variation in terms of physical appearance, expression of genes, behaviors that are genetic and inherited, and so on and so forth. So in general, the more variation we have, right, the more beneficial that becomes for any population or species, and the more likely some individuals get to survive for a longer period of time. In general, it's going to be much more beneficial for any living thing to exist in a population with a wide variety of phenotypes, okay? It's going to be a much safer kind of scenario. So when we think about variation among phenotypes, we can say that variation is stored in what we call a gene pool. And this is not a pool that you go swimming in the summertime. A gene pool is really just representative of all of the alleles found in all the individuals that make up a population. We can form new alleles when we create offspring, but if individuals move into or leave a location, they're going to alter the gene pool, okay? Different alleles within a gene pool, we can say, will exist at specific rates or frequencies, which is essentially a way of measuring how common an allele is in a given population. So in other words, if we have a high frequency of the dominant allele being present, right, that would mean that a lot of individuals are going to show the dominant phenotype, whatever that is. If we have a high frequency of a recessive allele being expressed, then again, we're going to see a lot of recessive phenotypes based on that. And obviously, as environments change, these frequencies at which we see these alleles will also change in response to that over time. So if we look at this as an example, right, we can see we're looking at the frequency of alleles in some type of mammal, right? We've got brown fur and black fur. And you could physically go through and actually count exactly how many of each color we have. Now, if I just kind of glance at this, it looks to be pretty even. Okay, maybe a few more that are brown fur, but for the most part, they're pretty even in terms of, how many have black versus brown. But over time, what we could see happening with something like this is we could have that population over time adapting to some change in the environment where the allele for brown fur might become more common or vice versa, right? And as that happens, we can see the frequency of these traits kind of changing over time. So this is the actual count for this, okay? So if I have 50, which is what's represented here in the diagram that you just saw, 20 of them are black, 30 of them are brown, and we can see they're identifying for us here that black is actually the dominant trait and brown is going to be recessive. In 100 alleles, we can kind of extrapolate that in a way and say, if this is true for 50, well, if I double those numbers, right, I end up with 40% or 40 of 100 that have the black trait and 60% that have the brown trait. So based on where this population stands right now, it seems to be that the brown is a little bit more beneficial, but it's not super significant, right? They're still pretty similar in terms of their numbers. But again, all of that can change as environments change. So when we think about this variation in the frequency of these different traits and the alleles that we're watching here, there are ways that we can kind of assess where this variation might have come from. And so there's 3 different methods where variation can be created. And the first is random mutation, which is probably actually going to be the most commonly seen. So mutations in general, when you hear that word, if you hear mutate or mutant or mutation, we tend to think of bad things. But in reality, all the variation we see among any species and population is coming from the accumulation of these random mutations. And these mutations happen in a cell when the cell's going to prepare for cell division and it goes to actually copy its DNA. Sometimes we make mistakes in that copying process. It's kind of like, if you're typing something and you make mistakes. Except the problem is in our cells, we don't have a great autocorrect system. All right. So sometimes those random mutations get passed down to daughter cells. And then over time, those daughter cells can be passed on and on and on. Or if it happens in a gamete, like sperm or egg, then you run the potential of passing it down to offspring. So over time, those random mutations will happen. Sometimes they're helpful. Sometimes they're harmful. Sometimes they have no effect. Largely, they have no effect. Okay. But if it turns out to be helpful, we can actually have a better likelihood of surviving in that population at that specific time. Okay. So random mutation is one way we can get variation. Another is called genetic recombination, or what we know as crossing over, which happens in the production of gametes. And for environmental science folks, what this basically means is that we're taking chromosomes and we're mixing up the genes that we find in those chromosomes. Okay, it's a whole molecular process that you don't need to be familiar with at this point, but we're taking pieces of chromosomes and we're basically rearranging them and mixing them up. So if you can imagine, we're kind of changing a lot at once, and that can lead to large-scale changes in the offspring that are produced from those gametes that are made in this process, from the sperm and egg that eventually will join to create offspring. If you're in biology, you should know something about crossing over. You should still remember that term. So go back to, if you need to, chapter 12 and look at that. But really, we're talking about, again, those chromosome segments exchanging information and creating new combinations of genes, okay? Lateral gene transfer is this additional thing that only happens in prokaryotes. So remember, those are just bacteria and nothing more. And they have this really unique way of forming these little tiny bridges almost, or tunnels, little passageways where they can take, if you have 2 bacterial cells sitting near each other and they form this little connection, DNA can be passed from one cell to the other through that connective piece. And so it's a way for bacteria to have a unique means of adapting more quickly to environmental changes. Okay, but eukaryotes can't do that process. All right, so single gene traits. One of the things we talked about a while ago, if you're in bio, is looking at just one single gene and how inheritance of that gene can be tracked over time and predicted. So we know single gene traits are simple, right? When we compare them to other things, these are the ones that are easier to follow, and they're certainly easier to calculate in terms of frequencies of phenotypes. And we're looking at here examples of snails. So we have snails with a phenotype with bands, snails without bands. And so if you were actually studying this characteristic and the frequency of this, you would go through and actually just count how many in a given population have that trait. And then from that, calculate a frequency or a percentage, right? How common is that? So based on this, it looks like it's much more common to be without bands than it is to be with, all right? Sorry, much less common to be without than it is. It's more common to have bands in this case. I think I misspoke. And so if you look at this environment, it's probably because it provides a little bit more camouflage and allows these things to blend in a little bit better. The harder types of traits to actually track in a scenario where we're looking at how genes influence natural selection would be polygenic traits. So polygenic traits are traits controlled by multiple genes. So that means we have many locations on many chromosomes that together all interact to create a phenotype, okay? It's not just one thing. It's a number of different sequences in our chromosomes. And so when we talk about polygenic traits, these are more complicated because you got to consider multiple factors. We tend to see more variation with traits that are polygenic compared to single gene, like the snails that we just saw. Either they have bands or they don't. But here in polygenic traits, we get more variation. We get kind of a more uniqueness as part of this, right? There are more intermediate types of traits that are in between the extremes. And so we can track the changes in those phenotypes over time, but it just becomes a little bit more complicated to relate it to a specific gene sequence. But one great example of a polygenic trait is skin color in humans. And, we know that with these types of patterns, we can see, here's darker skin colors over here, or the darkest, I should say, the lightest skin colors over here. And a lot of individuals kind of sit here in the middle of this. So this is looking at phenotypes, light skin to dark skin, and then the frequency here on the Y. And so if you consider kind of going along this pathway, well, the majority of us kind of sit in this average area, right? And that remains to be true for most types of polygenic traits. We tend to kind of sit in the average, and you will have extremes on either side. And you can think about height as an example. You could think about weight as an example, that both of those have genetic components. Skin color is one, right? Eye color could be one, but there is a range of traits, okay? A range of physical appearances for that specific trait, okay? So over time, these things can change. And as a result of that, that's where we can kind of track changes, like I mentioned. Okay, so you just saw I clicked on, and I didn't mean to do that. I wanted to see where we were at, but this is the end for 18.1. So if you've got any questions, let me know. Otherwise, we will talk more about it later, and I will see y'all later. Bye.